Ladies and gentlemen, we will now move on to the session 1 plenaries. I cordially invite Professor Sheila Nathan, Secretary General, FAOBMB, to chair the first session. Over to you, Madam.
Thank you, Ashwini. Good morning, everyone. So, as highlighted in the inauguration ceremony this morning, the COVID-19 pandemic has raised significant challenges for the higher education community worldwide. In particular, the urgent and unexpected need for face-to-face university courses to be taught online at the start of the pandemic. A significant issue for most of us at the universities at the start was the lack of online teaching and learning experiences, as well as the availability of tools and materials. So in this morning's session, we are delighted to have with us experts in teaching and learning of biochemistry and molecular biology to tell us how they developed and utilized innovative and creative techniques in the new normal. And before I invite the first speaker, I would like to inform you that there will be no question and answer session at the end of each talk. Participants are asked to type in their questions into the Q&A box, which you will find at the bottom of your Zoom screen, and to indicate which speaker they are addressing their question to. Your questions will be discussed during the parallel session, and the answers will be incorporated into the symposium report that you will receive once it is available. Okay, so let's proceed with our first speaker this morning, Professor Gabriel Hornig from Brazil. Please allow me to introduce Professor Hornig to you. He graduated with a PhD in science from the University of Campinas in Brazil. And having a keen interest in education, he undertook postdoctoral work on innovation and development of teaching and learning at the Institute of Education, University of Minho in Portugal. He's currently a professor at the Federal University of Alfenas in Brazil, and is heavily involved in the area of teacher training for the use of IT in teaching and in the development and evaluation of digital educational technologies for classroom and distance learning. This morning, Professor Hornig will be talking about conducting laboratory activities during and after COVID-19. May I welcome Professor Hornig?
Hello, thank you for this invitation. It's very nice to be here in this morning. In Brazil, it's 15 minutes to midnight, so it's a little different time. But good morning to all participants of this event. I'm Professor Gabriel Hornig. I work as a professor of biochemistry and educational technologies at Federal University of Alfenas in the state of Minas Gerais, Brazil. I thank the organization for the invitation to participate in this important event, and I believe that initiatives like this will increase, enhance the area of education in biochemistry and molecular biology. I also believe that there are global motivations that drive us to create networks of sharing and collaborations. Well, as I'm from biochemistry area, I like to start my presentation with a little pathway. This plenary lecture will address the principle for online teaching, presenting the case of adaptation of biochemistry discipline in the Brazilian public university, highlighting activities that can be carried out with a focus on laboratory practical activities using animations, simulations, and interactive videos embedded in the teaching platform. At the end, I will present very shortly the student authorship as a new possibility to teach and learning process. It's important to understand a little of the history of technologies in education. We start with more emphasis in the 90s with the use of tutorial applications, made like in toolbook instructor, and until now, at this time in low resolution, not just now, but on the way to more interactive system, which were boosted with the use of systems such Macromedia and teaching platform Moodle, and here in Brazil, Teleduc. Animations and simulations in Flash began to be popularized in internet in 2007. In a very short time, Java and later HTML5. With the populations of smartphones, Mobile apps are starting to take place, the place of desktop apps, include the possibility of collaborative builds in the cloud, from videos, concept maps to apps. And 2020, so the COVID-19 pandemic took most of us by surprise, demanded a rethinking of our class, the use of technologies developed until then, and in many cases, little used, and how to transform our classes biochemistry and molecular practice is this talk. The period of pandemic demand from us an improvement on our educational planning, in many cases forgotten, and we need more than ever to plan well. So for this, ask ourselves, what educational objectives of the course? What skills do you want to stimulate, develop? What are the difficulties in teaching and learning? Will it use own resource or not? How you make the technology available for your students, which strategy to use, and how to evaluate. And all this planning, mediated by technologies, require us to have an integrative view of their use, connecting affective, cognitive, and social aspects in our remote educational practice. As an example of curricular unit that underwent transformation during the pandemic, I present the simplified structure of basic biochemistry, which was based on active methodologies. The subject was organized in units and this in learning cycles. It started with the principle of flipped classroom. At this asynchronous moment, students had access to unit objective, various resources like tutorials, ebooks, video lectures, games, direct studies, educational software, and make a conceptual maps in the cloud, in the software that called CMap Cloud in groups. In the first synchronous moments, students receive problems, challenges, and other activities. So for each unit it was different. So sometimes based in problem-based learning, sometimes team-based learning, sometimes challenge-based learning. So for each unit was different. And discussion in resolutions, and the resolution of the problems in Zoom groups. In the second synchronous moment, the practical class of the week was oriented and discussed with the students. Sequentially, students developed practical virtual class using different resources for each type of class: interactive videos, bioinformatic resource, animation simulations. And during the units, students developed the concept map in the cloud as a teaching learning strategy and also as assessment instrument. At all the time, students had channels to ask questions synchronous and asynchronous with the teacher and instructors. A little about the assessment. To check the frequency of activity, the Moodle progress report was used. This is this bar here, green, blue, and red, so the students can see how is the progress. A formative assessment was planned. So all the time the students had feedback. So with individual exercise, with consultations, automatic feedback, guided students with the instructors, the concept maps, they post the concept maps embed in the Moodle forum and discuss all this. So you have enzymes, maps, problems about metabolism, about disease, put inside in the forum and discuss with the students. It was also proposed to build thematic seminars in addition to a final exam. Here are some of the materials used during the course, all incorporated or linked in Moodle. In the sense, I highlight the importance on the convergence of all resources for the teaching platform, increasing usability for the students. Among the resources used are, for example, games with augmented reality, crossword puzzles. I have here a game that's called As Aventuras de Kreber. It's like a Kreber Adventures. So it's a joke with crabs. It's very nice, and it was made by students. It's an arcade game. And so a lot of different kinds of materials. Well, going to the practical laboratory activities. Regarding practical classes, the practical laboratory activities are presented as an important curricular component for teaching proposal of biochemistry and molecular biology, from which students can develop scientific thinking, so understand and apply knowledge covered in the curricular unit. One of the challenges for teachers to teach online during the period of pandemic was to develop these activities effectively through the internet. In the photo here, you see students in laboratory during a course of beer science. So they are doing beer here. So it's very difficult to make beer online. And so when we think about laboratory activities, it's important to highlight some possibilities. The virtual laboratories with animations, interactive animations, and simulations. The remote laboratory. This is a real laboratory, but accessed and manipulated remotely. I have a friend, Eduardo Gallenbeck, from University of State of Campinas, that made a lot of these kinds of remote laboratories. And the laboratory in video class or interactive video class. Each of these has benefits, limitations, and each teacher needs to access what they are able to use and what meets with the objectives of the course. And when we think about this practice, it's important to be clear about what we want as the general objectives of our practice. We can be a mixture of motivational, practice, training, inductive, investigative. And this later, investigative, is more interesting, being greater with greater complexity. However, more complex to be performed online. And here a little about the potential of virtual practice. It's understood that there are benefits and limitations in these virtual activities when compared to face-to-face activities, like the experience of work in laboratory, psychomotor development, and other skills. But the online lab activities can provide flexibility of use, self-paced, as well the possibility of repetition and automatic feedbacks, depending on each application. It's another word that these methodologies can be part of the student's formative assessment process, enable feedback opportunities in difference from the traditional ones. In this case of presentation, we made 15 laboratory practical classes for the biochemistry course, built in the instrument in the Moodle that we call lessons. So we use lessons in Moodle. In each case, different resources were used, highlighting recording real classes incorporated in interactive system and use of simulations and bioinformatics tools. I will highlight here 2 of this practice, the buffer system and 5 virtual protein quantification, and then briefly introduce some features that you can use. For each practical class, experimental script, the complementary material, and virtual practice itself are presented. After the students answer questions related to the concepts discussed and interpretations of the result to have the indication of complete activity in the Moodle system. Here you can see for the practice of buffer system, the experiment was recorded, uploaded on YouTube, and linked in a software that we call Edpuzzle. On Edpuzzle browser software, you can add an interaction during the video, such notes, questions about the contents, techniques, interpretations about the results. One of the advantages of the system is being able to record students' interactions on Edpuzzle, including performance in the question, in order to access the class responses later. In addition, it has a low production cost and is easy for a student to use. So you can use this resource that is online because we know that it's very expensive and it would take time to make a new simulations for the students. I will change here my share. You can see here is in the Moodle, this video is embedded in the system. So I use here HTML code. So the students here, you can see these points here. So we have notes, we have questions that students can answer. And after you have another question to students. This is the more cheaper, it's more easy to do, but that is not so much interesting for the students. So let's come back. Well, so we changed a little about the practice. So we have now here a simulation. This software I made that called ProtVirt is for protein quantification. This is a support tool to biochemistry practical class. It's a free software, so you can download free here. Is open. is an interesting proposal for practical classes. In the ProtVirt software, you can find basic concepts about peptide bonds, biorate reaction, spectrophotometry, and highlight the simulation of the protein quantification by visible light spectrophotometer. Here you can see a little more about these simulations. It's very interesting that this spectrophotometer presented is similar to one that we use on the models in the practical class. And we have some operators, errors, and so students can simulate here. So I will open this software so you can have a little idea about this simulation. So here we can find different kinds of sections. You can use the mouse here. You can open this in Windows system or in a Macintosh system. So you can make like a little reactions. We have this in English and also in Portuguese. So here is more information, not so much interactive. So the spectrophotometer here, you can manipulate this, open the system, and is at the deck so you can see the light. And so you go to the protein section, so you can use here, prepare. So we made all the steps. So students can prepare the tubes. It's very interesting because I used this software before the pandemic to prepare students to go to laboratory. So when we think a hybrid model, you can use like these simulations to prepare students to understand spectrophotometry. And so after when we go to laboratory classes in the presence, we can make more investigative classes. So students can have some problems and using these techniques and other techniques to find these resolutions. So here is a little video. So we go, and I can here with these tubes, but here, and so in water bed 37 degrees. So I prepare the sample tube. And now I need to set the machine. We need to turn on. So let's put the wavelength. And if I try to make a blank, there is an error. Please note that the sample compartment is open. This is very normal for my students. Forgotten to open the equipment. So let's close. Oh, let's close without. So try again and insert the cuvette. So there are some errors that is inside the system. So can simulate some error in the laboratory. So now I have 0. And so the student now can put one by one. So this is one of the model of spectrophotometer. There are another that you can put another, not another, but 4 or 5 cuvettes in the same time. And the students make the measure for all the cuvettes. I mean, so only 2. And after this, they take all the data and they need to make the standard curve so they can use the LibreOffice, Excel, or Google Drive, I don't know, and find the correlation coefficient and the linear regression so they can calculate the cuvette 9 and 10. So they're not the answer here. They need to take all the data and come back to the software and make something, the formula to find the results. So let's go to another software that I use in my virtual class for laboratory. This is made by Unicamp, by Eduardo Gallenbeck and all the team. It's also free, so you can download here. It's a simulation for enzyme kinetics. So it's 3 experiments that you can use. One about the reaction speed, so reaction kinetics, another about competitive inhibition, and another about non-competitive inhibition. And the students can interact with the laboratory instruments and obtain the data in the tables and graphs for discussions and conclusions. So it's very interesting to use this application too. This is another software, very nice, because they have a lot of kinds of materials, contents, graphs, images, and simulations that call enzyme software. This software have some enzymatic reaction simulations that possibility the change like the pH, temperature, and concentration of the medium substrate. I'll try here to open the software here. Okay. So this is the enzyme software. We have two kinds of sections here about structure and function and about kinetics. So you can use here about text, images, animations, and simulations. So a student can navigate because we need to think that the student can have different learning styles. Some students need more visual, some students need more text, some students are more abstract, some students are more practical. So in this software, the student can navigate by different kinds of resource. So here we have a like simulation experiment. It's in Portuguese, but I think I almost can understand here like a pepsin, trypsin, polymerase. So let's put here the pepsin. And I have here pH, so I can change the pH for each number I want. So the student can try different kinds of variables, like I can change pH, temperature, and see what happens. And after we can discuss why they have more or less product, what can I make to make greater. So I see, oh, I can put down here, I can put down here. And so they can try different kinds of experiments. Like here, see the two graphs, so you can have a comparisons to see. So it's another kind of simulations, very interesting to the students. It's not the same like I talked the laboratory, but you can have something that you can do with the students. So let's come back to the presentation. It is another that I use. I remember going to running in my graduation, running track during my graduation to do exercise and take glucose and lactate and levels and discussing the data about this. So a shorter running, long running, and this interactive animations is a very simplified way allows to choose some types of exercise or types of fasting and simulate the use of some resource for ATP production, indicating the molecules used in the type and the type of metabolism for ATP product involved. Very nice. This is more technical also. This is for chromatography. This is a simulation of laboratory for performing ascending chromatography on paper. The students can interact step by step on the experiments, helping him to understand the execution and principles of the technique. This is made also by Brazilian universities of Rio Grande do Sul, Federal University of Rio Grande do Sul. So you can select the group of equipment, use the mouse, and begin all the steps. Is very nice, and also download here. It's only in Portuguese. All in another time they can make in English also. Finally, a last software that I want to discuss with all the people here is oxygen consumption by mitochondria. Is a simulation that can be performed on Windows, Mac, or mobile devices, involving the consumption of oxygen by mitochondria from insertion of substrate and diverse inhibitors and membrane decoupler. So graphs and oxygen consumption of the membrane can run over the time as components are inserted in the system. It's able to generate a very large of numbers of combinations from which the result can be discussed and help to understand the production of ATP by mitochondria. So let's go open and share a little about this software. So here you can see the image of real experiment. So in B here you have the incubation cell. So the membrane in number A, electrode, and here I can change the color and I begin here putting the potential graph. I can put mitochondria. So begin to use oxygen. and I can change the speed here. So let's go put pyruvate, and I begin to use, and the membrane potential grow up. So I can put more zinc, and I can see here the molecule, like here, I can see all the molecules and have some informations. So it's very nice. I will put here succinate. So more quickly, I use the oxygen more quickly, so I can ask the students why, when I put pyruvate, I start to use oxygen, and after when I put succinate, it's more faster. And I put here like 2,4-DNP, so as the coupler. So let's see, the use of oxygen is very, very quickly, and the membrane potential go down. And I can ask the student why. So I can make a lot of kinds of combinations here and make the students to think. And when the student use it on mobile apps, here you can have a little animations that they can click and see what happens, like in some structure models. So it's interesting to make. So, well, it's understandable that few teams and professors of biochemistry and molecular biology are able to produce their own materials for material class. Like I have a team that can make some materials like the Protivirt and others. I have some friends like Eduardo Gallenbeck and Vera Tristenbrad in Brazil that make also, but it's there is a significant numbers of materials freely available on the internet. So below, I will indicate very shortly some sites that you can find some interesting resource for you. One of the sites is a remote labs and simulations, and I will show two examples of resource below. One is an Excel spreadsheet based on HPLC simulations, allowing you to explore several laboratory variables. It's very nice. This is a UV visible and fluorescent light spectrum simulation. So all this you can download or use on web. We have also, this is very known, the Fat Colorade's websites have a lot of resources. This is two resources, one about pH and measurement, and another of salt solubilization. This is the MIT open course website, where several parts are also available, such as titration. All this will have the link here, so you can record this, share this screen. And finally, the Lab Exchange website, which has several features such as simulation of gel electrophoresis. And the students will be able to manipulate the equipment and reagents, so you can put the cable, turn on the equipment, make all the procedure for the electrophoresis, and perform all the procedure. Well, there are many resources available on the internet, as well as new teaching and learning methodologies and possibilities. To leave a taste of one to more, I present a possibility that I have been used in biochemistry curricular unit and in university extension program. The students themselves build their teaching material as a group. So using a block program language that we call Blockly, like using MIT App Inventor or Scratch application and other, is 100% free. The proposal can be varied, like teams about disease, experimental techniques, what you want. In addition to developing knowledge of biochemistry, they develop digital skills, social affective skills, and logical and mathematical reasoning skills. And these reasoning skills are extremely important for learning biochemistry. So like metabolism content is all connections. So when students develop this reason, help to them understand metabolic contents. So I use this with students for the first years from the university, making softwares to Android, to mobile device, about disease, about biochemistry, and they like so much, and they think in some resource that I made that they learn when they construct these applications. So let's see what happened to my screen. Okay. So you can see here like the blocks from programmation, and you can find a lot of resource like here in Portuguese, in English, in EduCaps. This is a digital library of science that I help to in the maintains and another websites. And once again, I thank the organization committee for the invitation to present at this important event. And I hope I could have contributed a little for you to reflect on your remote virtual classes in this theme of pandemic and post-pandemic. Thanks also for the audience for their interest in the area of education and participation in this plenary lecture. I indicate my contact here if anyone wants to discuss new ideas, suggestions, collaborations. I will be online for some networking. And you can also download the presentation with the QR code, so you can take this screen and see after this presentation again. So that is.
Thank you very much, Professor Horning. That was an excellent presentation, and I'm sure a lot of us would like to adopt your approach to translating face-to-face lab practicals into online practicals. In my university in Malaysia, we are still trying to figure that out. So I'd just like to remind all the participants, if you have questions, to please type them into the Q&A box. Okay, in the meantime, on behalf of the organizing committee, I would like to present Professor Gabriel Horning with a certificate of appreciation. So as it states here, the certificate is awarded to Professor Gabriel Horning in appreciation of his contribution as a plenary speaker during the IUMBMB FAOBMB CBSL Virtual Education Symposium 2021. Thank you so much.
Thank you.
Our next speaker is Professor Glenda Gillespie. Professor Gillespie obtained her Bachelor of Science degree in biology from Auburn University and went on to earn her PhD in biochemistry from Case Western Reserve University in the US. She did a postdoctoral fellowship in plant biology at the University of California, Berkeley, and from 1998 has been affiliated with the Department of Biochemistry at Virginia Tech, where she is currently a professor and department head. The focus of her research is on the molecular pathways that plants use to respond to the environment, and also on the development of a genome scale model for Arabidopsis. Her group is involved in a number of outreach projects related to education at the school level. And today, Professor Gillespie will talk about best practices for taking experiential learning online. Professor Gillespie.
Thank you. Let me share my screen. Okay, great. Thank you very much. It's my pleasure to be here and tell you about 10 best practices that we put together about concerning an experiential learning process that we took online last year. And I just want to start by acknowledging my graduate student, Caitlin Cridlin, and my collaborators, Tiffany Drape, who's a social scientist, and Sasha Marine, who's also a faculty member in the Department of Biochemistry at Virginia Tech. This is a project that's funded by the United States Department of Agriculture, a specific part called NEFA, and it's a summer undergraduate experiential learning project. I've got our social media contacts here, and I've got them on my last slide as well. And so please don't hesitate to reach out if you have any questions that I don't get to answering tonight. Well, it's tonight here, this morning for most of you. So let me give you a quick overview of our summer undergraduate research program. And it's focused on plants. I'm a plant biochemist, and it prepares students to bridge the gap between basic science and applied science. And in particular, we're really interested in how students can, the next generation of plant scientists can be trained in both discovery and how to apply that to solve real world problems. And this is actually a photograph of a cornfield in Virginia. I'm going to show you a little bit about where the project takes place. But first, this is inspired by lots of different types of publications. And there's one specific here called Feeding 9 Billion People, the Issues Facing Global Agriculture. And our project really consists of 4 parts. The two halves that are important for my talk tonight are the focus to train research. Okay, so these are students who finished at least a year of college, and then to also give them the proper professional development so that we can really train students to enter the workforce and be very creative, be trained both to understand molecular processes, biochemistry and molecular biology, but then how to apply that to practical problems. So here's where the project takes place. I'm showing you part of North America. And if you look on the right here, you can see these are the Appalachian Mountains that are close to the eastern coast of the United States. The institution I'm at, Virginia Tech, is located where the star is, and you can see this aerial view of the campus with the Appalachian Mountains in the background. So I'm going to zoom in on the state of Virginia. So the state of Virginia is located over here on the East Coast, but Blacksburg, where I'm located, is in the southwest corner of Virginia. And Virginia has, you probably know it as being close to Washington, D.C., but for those of us who live there, Virginia is also has a lot of rural farmland. And as a land grant institution, Virginia Tech is focused on having research stations within those farmlands. And so what I'm showing in these other yellow dots are our partners in this project. They're Agriculture Research Extension Centers, or ARECs, where Virginia Tech scientists, faculty members work on practical applied problems that are embedded within the living learning laboratories of rural Virginia. And so we have another partner on this project shown in these blue stars, which are several different, sometimes minority serving institutions like West Virginia State and Virginia State. And these institutions send students to our summer programs, and that helps us with our diversity and inclusion goals. Okay, so the really key about this program, it's 10 weeks long, and students would normally, pre-pandemic, spend half the summer, 5 weeks, in a basic science lab in Blacksburg doing molecular biology and biochemistry. And then they'd spend the second half of the summer at one of those AREC stations. And this is a picture of a field with a drone. And so by getting these two disparate experiences that often are linked by a shared research problem, we're hoping to train students in a new and different way. And this is a picture of our fellows from summer of 2019. And just once again, this is a collaborative project, and normally students would be in two locations, Blacksburg and one of the other stations, and then they drive back to Blacksburg and present their results at the end of the summer. So we have a really interesting mentoring structure, and we collect data. I'm not going to talk about the data. We haven't really collected enough to have an overall picture, but I do just want to mention in a normal year, our undergraduate researchers would have a research faculty member on campus and also a graduate student who were focused on mentoring that student for half the summer. And then they would have another faculty member at our research station and another graduate student, so on campus and off campus. So they have a really interesting mentoring network. So now I'm going to talk about how we transition this to online. And I think this picture really kind of summarizes the chief problem, because as we all know, here we're looking at a student and we're looking at a mentor in the greenhouse. This is from that applied part of the project. And it's really that one-on-one working, hands-on, the apprenticeship model. That's how we teach students how to become scientists. And so, of course, that became transition to this process, which you're all familiar with, which is Zoom. And so I'm just going to point out, this is our graduate teaching scholar, my PhD student, Caitlin Cridlin, Sasha Marine, who directs the program. This is our undergraduate biochemistry advisor, who helps us by meeting with students, checking in on them. And then I have the 7 fellows or students that were part of the program in summer of 2020, and also some alumni that came back and visited us. And this is one of our Friday sessions. So that's the way the summer looked. And so now what I want to do is describe how we overcame one of the biggest hurdles, which was how do you do experiential learning, research, authentic inquiry, and do that virtually at a distance. So virtual mentorship, and this is really just the basic science portion of the project. I don't have time to talk about the applied research that students did, but it used a pretty similar model. And what we decided to do was to use this outreach project that my lab has been participating in for almost a decade. And it's called the Partnership for Research and Education in Plants, or PREP. It involves high school students in authentic research. My lab is a collaborating partner, and the author of this and originator is Dr. Aaron Dolan, who's now at the University of Georgia. And so in this project, students work with mutants from my lab, and I work on Arabidopsis thaliana as a model system, and we have many different genetic mutants where genes are knocked out for genes that encode specific enzymes in the signaling pathway we study. And in the high school classroom, this is used to illustrate how genes have different functions. And what I'm showing is one of our visits to a high school classroom where students are growing 4 different pots of plants. The green tape, these are the mutants, these are the wild type. And usually what they are doing is designing an experiment to uncover gene function. And it most likely involves creating a stress or a different condition. And so they design the experiment, they create a hypothesis, and then they collect data. And at the end of the project, in a high school classroom, pre-pandemic, we would go and visit, and the high school students would talk to us about their data collection, and we would talk about how one draws conclusions. So we decided to use that and create a hands-on research project. And to do so, we mailed out kits. So we took seed from a series of mutants and wild types because these were college students, so we wanted something a little bit more sophisticated. We sent them soil, pots, lights, and then we sent them anything else that they needed along the way. And their very first box came, and this is a picture of Caitlin doing a tutorial on what to do with your box. So we actually had an unboxing day. This is a Zoom snapshot, so all of the interactions occurred virtually. This is Janet Donahue, a technician in my lab, and once again, Sasha Marine, and a couple of different students. The other half of this was to do what we called our big data project. And I'm going to tell you a little bit more about that in a different slide. So I just want to summarize that, give you some examples of the student work. And so this was the end product of one student who asked, does varying the amount of these signaling molecules in inositol pyrophosphates affect salt tolerance in Arabidopsis? They collected really nice data with our help through virtual sessions. And they learned how to manipulate data, and they drew conclusions and did a presentation in the summer. And this is a separate student's work who decided to look at high humidity. So once again, the impetus is on the student to come up with a hypothesis, to think about what would be interesting, and then to take that further and set up conditions to collect data. So now I'm just going to share with you 10 best practices from how we implemented this and what we learned. So one of the first things was we found that students need a clear structure and they need objectives for the virtual program. So we used a learning management system that's shown here. We used Google Docs. Everything was linked on this site. The students could go to this site and see the schedule for every day. They could click on every single Zoom link that we use for the entire summer. And so that was helped a student navigate where they should be in the virtual world. The other point I want to mention is this is just a calendar for one representative week. It was very important to keep students engaged and give them the structure throughout the week. So for example, we always started off the week with a Monday 9 a.m. meeting. And because this was centered around my lab's work, all members of my lab formed the mentoring team. And so students came to our lab meeting Monday mornings at 9 a.m. and they heard my lab group talk about their research and what they were doing. We were back in the lab at that point because the project started in mid-May. And so we had the ability to be in the lab somewhat last summer. Then at noon on the same day, our student fellows came in, and as the summer progressed, they had their own data and they presented that to my lab. On Tuesdays, we had Journal Club, which Sasha Marine, she implemented that, and it was based on a course she teaches at Virginia Tech. And then we had individual meetings with students on Wednesdays and Thursdays. We had a scientific workshop, a different professional development event every Wednesday, And then on Friday, we had what we called checking in and professional development workshops. So giving them programming, so they had about 7.5 hours a week on Zoom with us in either lab meetings, journal clubs, or professional development activities. And then it left them about 25 hours a week to do the hands-on portion. The second thing I want to mention is Zoom and virtual etiquette. And I think probably most of our students, at least in the US now, know how to use Zoom because that's been the preferential platform. And there are a bunch of new platforms being created. But anytime you use a platform and you've got college students, you should take them through how you want them to use that. And the other thing you should do is talk about virtual etiquette. And this is just a graphic from R.L. Clooney's site, but it has several of the things that were important to us that we addressed right up front. Muting, whether or not we expected the camera to be on. And we did for some things and not for other things, but we expected students to be engaged. We expected them to be respectful of everyone's differences. So don't forget to go over those things. We also tried to find out what concerned students, and we did that at the very beginning of the summer. And I'm not going to go through all of the findings from this. I'm just going to highlight this one under communication. A student said, if we have a problem, it might take longer to ask questions and get answers through email than it would in person. Now, that's something they said at the very beginning of the project. And so based on that, we decided we're going to have these individual meetings with students. So if a concern is how the communication process will work, and it might be more difficult for some students than others, then we'll set aside an hour each week where one or more people from the lab are connected to Zoom to that student individually. And that was an important thing to do. Number 4, create a mentoring network. And so I've mentioned this a little bit. We had the scientist faculty member who's directing the program, the graduate teaching scholar, we had our undergraduate student advisor, we had myself, and we had 3 other people from my lab as well, a research scientist, a technician, and an advanced graduate student. We were the mentoring network, and we were highly engaged throughout the summer. We were also the experts on the scientific questions that they were asking and answering. The other thing I want to mention are these alumni again. This turned out to be a very powerful mechanism. We brought back students from previous years on a Friday social event so that they could talk to the students about how our program had impacted their careers. This person now works in the biotech industry. This person and this person are actually in PhD programs. And this is Chase, the biologist, who's a scientific communicator. And so this is a really great thing that we've continued to do in the project, which is bring back alumni because it's easy to do on Zoom. Number 5, use synchronous tutorials. And what I'm showing is a picture of our unboxing day where my technician, Janet, is taking the students through what to do with all the materials we sent them. We had a lot of safety training. We also sent a letter to parents or guardians of where the students were going to be present so that they understood the safety concerns and how we were going to mitigate the use of these genetic mutants. And the bottom line is we mailed things to students, and before they got their last stipend check, they had to mail everything back to us. And they all did, and it worked really well. Caitlin is here in our Arabidopsis potting room and could use her camera to go through the exact same process that the students did in real time. The students could also use their camera or their cell phone to show pictures when they were confused about whether or not their setup was working. And this is just an example of a little protocol slide that Janet put together. And one interesting thing is Caitlin had found an app online that can convert your cell phone to a kind of a rudimentary light meter. And so these were the sorts of like tutorials we did that were extremely impactful and kept students engaged and also engaged in the human element of research. So, and I think our keynote speaker mentioned this, community is really important. And it, of course, is one of the hardest things to do in a totally virtual environment. So Caitlin had set up a few activities, and pretty soon we realized they were building community. So we had social events every Friday, and this is a picture, I think, from one version of what was named Fancy Dress Friday, which was come in your party clothes, and several people brought their favorite things to eat. Every other Friday, Fancy Dress Friday was come in professional attire, and then the students would talk about that professional attire and get some feedback on that. And these turned out to be fun, and actually in our assessment, students said they would have liked to have had more of these. And this was not something really at the beginning that we thought about, but we strongly encourage you in any kind of virtual experiential learning to make sure to include social events, because our students really did get to know one another, even though they never met us or one another face to face. So we leveraged a lot of online resources. The previous talk was just fantastic at giving lots of resources. I really appreciated that. I think, you know, those will just grow. And I just wanted to highlight, so students had to take, had to go in and leverage gene expression sets. So there are gene expression sets either directly from our mutants or related to some of the aspects that students were exploring. And so Caitlin designed a tutorial to take students coming from a variety of backgrounds all the way through how to extract that data, how to analyze that, how to use gene ontology, and then how to come up with some hypotheses based on what you're seeing in the data. And most of the students really enjoyed that flavor of big data. So number 8, we utilize workshops to supplement the training experiences. We did these in the face-to-face version, and we converted these to online, and they worked extremely well. I'm showing you two kind of slides from 2 different workshops. So this is Floyd Childress, who is a Virginia farmer, and he meets with our students, and normally they would tour the farm, and he talks about what traits are valuable and what sorts of technology he envisions would make farming even more environmentally sustainable or increase yield, decrease pathogens, et cetera. And so here he is meeting with students on Zoom, and that actually worked pretty well, and we got positive comments from students. They enjoyed meeting this farmer. This is another workshop that we have where Dr. Jacob Barney comes in and talks about how do you present your science, how do you make a PowerPoint presentation. And all of our students at the end of the summer had to record a video for our summer undergraduate research symposium. And so they really used these scientific communication workshops. We had one other workshop on how to communicate. So number 9, make time for one-on-one meetings. I've already mentioned this. This was probably one of the most highly impactful things. It doesn't quite replicate what you get in person, but it was a time not only for, I'm not sure my plants are going correctly, I don't understand this RNA-seq data, but it was also a time for connecting with students about a variety of things. I'll share with you that one of our students had a family member pass away of COVID during summer of 2020. That student really struggled. He finished the program, and so those one-on-one meetings gave him a little bit of a lifeline to some other humans that he could share some of his concerns with. So that's a very impactful practice. And the last one is leverage virtual conferences. Here we are in a virtual conference, and it's, for me, it's incredible to be here in the evening in Blacksburg, Virginia, and to hear all these great thoughts. Last summer was kind of the beginning of that, and the American Society of Plant Biology, ASPB, held a virtual conference, and they made it very accessible by having student prices and one-day admissions and group admissions. So it was financially very easy for us to buy admission for one day to all of our students to attend this world-class meeting. And so what we did is we asked the students to look at the program, plan out what they were going to do with their day. They knew that the meeting had multiple days, and they knew they were only going to attend on a certain day. And we asked them to ask questions. And it was incredible to attend this international meeting and then to see one of our summer fellows ask a question in real time to a speaker at this meeting, and it was a really great experience for them. They also did a presentation at the end of the summer on our campus, and this is just one of our students who was very interested in the impact of copper. And so, of course, she was looking at our mutants that have changes in inositol pyrophosphates. And this is just an example of her data. She grew plants, she did applications, collected data, and these were really excellent presentations. The students were able to carry out what we consider to be authentic inquiry and to really collect that data, put it together, and learn, even though they were in their homes, apartments, condominiums, or some of them were actually in dorm rooms. So that's what I have to tell you about best practices. And I just want to acknowledge again, Caitlin Cridlin, my PhD student and a graduate teaching scholar, Sasha Marine and Tiffany Drapes, and then the students in our program, which we call VT Real Fellows, because it was really a joy to work with them last summer and experience this. Once again, we're funded by the USDA NIFA, and we're supported by Virginia Tech. And here's our social media. Please feel free to reach out via social media or other means. And thank you very much for your attention.
Thank you so much, Professor Gillespie. If I could ask just one very quick question. Is it easy to do what you're doing with a much larger class?
So the question is, could we do that with more than 7 people? Could we do it with 25 people or 35 people? I think you could, because the time sink is those synchronous tutorials and the meetings. I think you'd have to have maybe a little bit more robust or more person power in the mentoring structure, so that it doesn't fall to 5 or 6 people. But if each of those persons did a couple one-on-one meetings a week, you'd get closer to the goal. So parts of the project are scalable. That's the short answer to your question.
Okay, thank you very much. So once again, let me just remind you, if you have questions, you can also contact the speakers directly. Okay, so thank you very much, Professor Gillespie. And again, on behalf of the organizing committee, we would like to present you with a certificate of appreciation for your role as a plenary speaker in today's symposium. Thank you so much.
Thank you.
So let's move on to the next speaker in this session, who is Professor Paul Craig. Professor Craig is a professor at the School of Chemistry and Material Sciences at Rochester Institute of Technology in New York. Following a postdoctoral fellowship on the biophysical chemistry of blood clotting and 5 years as an analytical biochemist at BioQuant, which is a biotechnology startup, he joined the Department of Chemistry at Rochester with plans to use computers to teach biochemistry. So his students have developed computer simulations of protein separation and software for molecular visualization and predicting protein function. He helped to found the BioMol that we heard about earlier, which promotes molecular visualization literacy. He currently leads the Biochemistry Authentic Scientific Inquiry Lab, or BASIL, with faculty members from more than 10 campuses where students use in silico and in vitro techniques to predict and test protein function. He's currently exploring the use of Jupyter notebooks and virtual reality in teaching and research, and will tell us more about this during his talk. So I welcome Professor Craig.
Well, thank you very much. It's an honor to be invited to speak here. Thank you for that gracious introduction. It's just very exciting for me to be part of this conference. Speaking to you from Rochester, New York in the United States. So I'm going to do my best to bring up my presentation and share my screen properly. Right. So The title of my talk is Python Scripting for Biochemistry and Molecular Biology Teaching and Research. And one of the reasons I'm here is because I actually, I knew Gracia from the conference in Manila in 2019, the education conference there, IUBMB. And Gracia and I both attended the Teaching Science with Big Data conference sponsored by the IUBMB and the ASBMB. Can you hear me?
Sorry, Professor, sorry to disrupt you. Just if you can put the screen on full, then, because we can see the presenter view at the moment.
Oh, okay. Let me stop share and start again.
You can just select the whole screen there. There you go.
I'm sorry, I picked the wrong screen. Yes, so I encountered Gracia at the Teaching Science with Big Data conference, and that really reinforces in my mind, part of the new normal, which is the idea that computational skills are going to be important for us and our students as we move forward in both teaching and research. In fact, I spent the earlier part of this year with Jessica Nash and T. Daniel Crawford at the Molecular Sciences Software Institute, which is located at Virginia Tech. It's almost ironic that the previous speaker is actually a faculty member at Virginia Tech. But I was working with them this previous 6 months or so. And so let me give you an overview of my talk. We're going to be talking about data analysis and teaching biochemistry. And kind of part of this is also my story of how I ended up going down this path. The first thing I'm going to talk about is Jupyter Notebooks. Jupyter, you notice it's not spelled quite the same as you would spell the planet Jupiter. The PY there refers to Python. The R on the end refers to the R statistical language, and there are a couple of other languages also referred to in that title. So Jupyter Notebooks are a place where you can write code, but you can also record text, and we'll take a look at a little bit of that as we later in the talk. I'm going to talk a little bit about data analysis and the limitations you have when you're working with Excel and other spreadsheets like Google Sheets or numbers. And then I'm going to talk about creating the Jupyter Notebooks and the workshop that I developed as a result of the time I spent with the folks at the Molecular Sciences Software Institute, or MolSI. And then I'm going to do a live demo of some of the lessons from the workshop. And then I'm going to wrap up by talking about future plans and identifying and highlighting some resources that are available. And I will also make my slides available, through the people who host this conference. So when I think about data analysis in a teaching lab, I'm focused particularly on two of the labs we have in the basal curriculum. Basal, once again, is biochemistry, authentic scientific inquiry lab, where we're using computational and wet lab methods to predict function for proteins of unknown function in the protein data bank, and then go in the wet lab and test our hypotheses. And so two of those labs are protein concentration and enzyme kinetics. And in those labs, we generate a lot of data. And so I have a desire for the students to learn more deeply, and they really do as well, things about their data that they can understand if they do the computations right. And so one of the things, for example, that's important to me is confidence intervals and what part of a calibration curve is the most reliable. And also the challenge of nonlinear curve fitting, which is something I've been staring at for, oh gosh, since I was a postdoc 35 years ago. So a colleague of mine at the Rochester Institute of Technology actually demonstrated Jupyter Notebooks to me about 3 years ago and showed me about simulating enzyme kinetics in a Jupyter Notebook. And subsequently, I actually attended a workshop offered by the Molecular Sciences Software Institute on Python scripting and computational molecular sciences. And that led to a professional leave that I spent with them earlier this year. So what is a Jupyter Notebook? A Jupyter Notebook is a notebook you can see here, and we'll be looking at a live demo of a Jupyter Notebook in just a few minutes. But you have you can write in text in a Jupyter Notebook, so you can record information just like you would in a written notebook or in a Google Doc or something like that. But you can also do calculations. You can assign variables. You can do Python Python programming and calculations there. And so why would somebody want to do Python scripting in Jupyter Notebooks? Well, the computational power exceeds Excel. So you can do a lot more, and you can customize it very easily in a Jupyter Notebook. They are flexible and customizable, so you can do what you want, and your students can actually explore new things and find new tools that are out there. There are remarkable resources available. So people like me and with much greater skill than I have in this area are producing resources and generating new libraries and new tools all the time. And these are available because this is an open source community. So the members of this community are creating these resources and they're freely available. Now, there are a number of barriers to change to moving toward using more computationally intensive processes in our classes. One of those barriers is you probably already have some very good resources, and you're probably comfortable with those resources. And so the question is, why should you want to change? And I'm hoping that looking at some of the demonstrations here and maybe participating in a future workshop would stimulate you to consider that. You may not have any experience with coding or very little experience with coding, and so you're thinking, oh gosh, I don't know what to do. And I just led a workshop about earlier this month, and there were many people in there that had almost no experience with coding, and afterwards they talked about ways that they would use coding in their teaching. There might be institutional structures that make your institution not very flexible to changing maybe the firewalls on the computers or the way they handle computer security prevents you from being able to do this. So you need to work with them to find new ways to make this possible. Also, maybe the textbooks you use include online resources and it's all built in already. And so you don't want to go outside the bounds, and that's something that works very well. Maybe you're in a multi-section course and you'd like to change what you do in your section, but there's pressure because there are maybe 5 or 6 or 20 sections of the course you're teaching, and everybody wants to have common exams. And so these are all different challenges you might come up against, and so these are barriers you'd have to face and address. So to select the topics for the notebook topics for my workshop, I started with the Python scripting from computational molecular sciences workshop that I took 2 years ago from MOLSI, and actually did pretty much use the first 3 lessons that they have and modified those for biochemical purposes in my workshops. I also, I was thinking about designing things, and I came up with a list of things I wanted to do, and then my wife suggested, well, why don't you ask your colleagues? You have lots of friends. So I interviewed 8 or 10 of my colleagues in biochemistry and molecular biology to ask them what is their experience with coding and with computing. And also, what would they like to be able to compute that they cannot compute right now? And they came up with some suggestions, things like raw data, converting their raw data they maybe get from a microplate reader into derivatized data that they can then use and analyze. Also, being able to make predictions of things like the amount of enzyme you need to do an assay. Being able to see, if you look at the pH activity profile of an enzyme, and we saw some of those earlier from a colleague from Brazil who presented, looking at those pH activity profiles, what would be the impact if I substituted or modified one of the amino acids that's involved in the active site of that enzyme? And then finally, looking at dynamic calculations, equilibrium calculations that are dynamic, and being able to modify those things in a way and look at multiple possible ways to understand those. So the workshop that I designed is actually designed to run in 4 3-hour sessions. It's a live coding workshop, and I'll explain more about live coding later in this talk. There's a focus here on practice rather than on the underlying theory. So you actually get hands-on tools that you can use. Now, if you're like me, you probably want to understand the theory. There's lots of great resources out there, textbooks, free online instruction, lots of YouTube videos, lots of really great stuff. But the purpose of the workshop is actually to give you something you can work with, take home and maybe put to work in your classroom. And then finally, as we went through, there were check-ins each day during the sessions. Check in to see, could you get the code to work for you? And also homework you could do overnight before the next session. And the workshop consists of 8 modules. There's an introduction. Modules, first, the next module looks at file parsing, looking at pulling information out of a PDB file. And the next module looks at pulling information from multiple PDB files, PDB, protein data bank, structural files, and then writing the data you extracted to your own files. We also look at working with pandas, which is a data management library in Python. We did some linear regression, looked at creating plots in Jupyter Notebooks. And then there are 2 modules on nonlinear regression, looking at Michaelis-Menten kinetics. And to do the workshop, I had to recruit participants. So the first place I went to the Teaching Science with Big Data conference. It was sponsored by IUMBMB and ASBMB. And I asked them I was asked to present, so I made a presentation there for about 15 minutes. And then I also appealed to them, if you're interested, to check in with me. And I used the social media resources that were part of the conference to connect with people. I also emailed contacts that I had through the BASIL process, or the BASIL curriculum, people that are using it. And finally, I just reached out to friends, people who had expressed an interest. And we ended up with about 30 participants that way. Now, the workshop, this particular workshop was scheduled, we ran it from 2 to 5 p.m. in the afternoon, and I was running it from the eastern part of the United States. And this included, it was set up this way so that people on the West Coast would be doing it in the morning, and people on the East Coast could be doing it in the afternoon. And ran it on every other day for 4 days, so a Wednesday, Friday, and then a Monday and a Wednesday. And that gave some time between sessions so that the participants could absorb what they had encountered, probably for the first time, and also work on the homework and see if they could do the exercises. The way we did the workshop was it was a virtual workshop over Zoom, and I was the presenter, and Jessica Nash, who was my preceptor and mentor at MOLSI, and Ashley Ringer McDonald, who actually taught the first workshop I attended and is a colleague in BASIL with me. They were co-hosts, and so they helped anybody that had trouble. They would help them catch up. And it was also important to have stable connections, which Zoom was pretty good for. And before starting the workshop, I sent out emails and instructions to participants so that they could install something called Anaconda, which enables you to run Jupyter Notebooks on Macintosh or Windows computers. And they also needed to create a GitHub account. GitHub is a software sharing tool. So this is actually a picture of Anaconda. So this is the Anaconda interface, and it has a lot of tools built in. And I'm going to focus on two tools that are shown here. And these tools are the Jupyter Lab and the Jupyter Notebook. So these are 2 interfaces to get in to work with Jupyter Notebooks. And in addition, as part of my time with MOLSI, I created a Jupyter book that's online, and I'll provide the link to that at the end of this presentation. And we'll actually take a look at this book, and this contains all the materials for the workshop. Now, the approach to teaching this workshop started with live coding. Live coding means that I start off with an empty Jupyter Notebook, just like the participants in the workshop, and we build our code 1 line at a time. And best practices are to explain the code and actually talk through everything that I put on the sheet so that people can understand where we're headed. And one of the things I learned, this was my first time offering the workshop earlier this month, and one of the things I learned was a need to explain what I'm doing, why I'm doing it, and how we're going to learn. So that what, why, how is something I attempted to reinforce every time. And as I mentioned, as host, I would lead the lessons, and then my co-hosts would help people whenever challenges came up. And we had regular checks for progress, and this was in Zoom. And so the way we did this was with some of the Zoom tools that you can use in Zoom. So for example, if we did an exercise, we wanted to check did people get it completed, did it work for them? If it worked, they could check the green check mark. If it didn't work, they could check the red X. Also, if they had a question, they could raise their hand. If they just wanted to indicate that something was going well for them, they could give a thumbs up, things of that sort. And if it was a face-to-face workshop, we'd actually have green and red Post-it notes so that the co-host could see what's going on. So now I'm going to move into a live demonstration of some of these notebooks, and we'll be looking at calculations in Jupyter Notebooks, at plotting the data, and at nonlinear regression of Michaelis-Menten data. So I'm going to try to transition to that screen. All right, hopefully now you're seeing my web browser, which contains the Jupyter book that I wrote on Python data and scripting for biochemists. And could somebody let me know, are you in fact seeing that screen?
No, Professor.
You're not, okay.
Look,
So I'm going to start. I'll start again, and I'll share it, and we'll bring it up.
Yes.
There, you're seeing it now. Very good. Thank you. All right, so this is the Jupyter book. It contains those 8 modules that I mentioned, plus a setup, a page you can go to learn how to set up Anaconda on your computer, and a workshop schedule, and then the 8 different modules. And if you look over here, you can see we've got some navigation through each of the pages. So it's very convenient. And if I go to Jupyter Books and I set up my I launch Jupyter Notebook, I get a screen that looks something like this, and I can go in here and then launch the notebooks I want to study. And so this is the introduction notebook, and I want to talk a little bit about the format we use for this. So each of these lessons starts off with an overview, which includes questions, 1 or more questions. What are the main things we're trying to answer in this lesson? So in this particular lesson, the question is, what is the basic syntax of the Python programming language? Syntax is like the grammar. That's how you write things in Python. And the objectives in this case were to learn to assign values to variables, to use the print function to check how the code is working, to use multiple assignment to assign several variables at once, and finally to use a for loop to perform a task repetitively, so to do the same action over and over again. And so I'm just going to go through and show a few things in the notebook. And so this is a coding cell. And in a coding cell, you can actually go in and do a calculation. So if I go in here and I say 17 times 441 and execute that cell, I get the product. So I can actually do simple calculations here, but that's really a pretty short list of what you can do. There's a lot more you can do than just do simple calculations in these coding cells. There's another type of cell in these Jupyter Notebooks called a markdown cell. This is a markdown cell where you put in simple text using something called Markdown, which is actually what people use to write in Wikipedia. And then when you execute those cells, it comes out looking nice. And in this is a code cell, and in this code cell, I assign a variable for Michaelis-Menten kinetics. So we have a Km of 15 micromolar, a Vmax value of 100 nanomoles per second, and a substrate concentration of 8 micromolar. And then we put in the equation for the Michaelis-Menten, or actually the Michaelis-Menten equation here. And if I execute this cell, it does those things, but I didn't tell it to print anything. So I didn't tell it to print the velocity. So if I go to the next cell and tell it to print velocity, it gives me the velocity. And I could create a fancier print statement that actually would give me the velocity with the correct number of significant digits and some information of units and things like that. And so it's possible not just to assign variables one at a time, but in this cell, I actually assign the variables, 3 variables here, the Km, the Vmax, and the substrate concentration. I assign the values to those here. I use the Michaelis-Menten equation again. When I print the velocity this time, notice the Km, the Vmax, and the substrate concentration are different. I get a different velocity. And so that's just very simple things you can do in these notebooks. And finally, I wanted to mention something about for loops. So a for loop is a tool you use to do a repetitive task. So this is somewhat analogous to being able to copy cells down in Excel. So if you are familiar with copy down in Excel, it's kind of like that, but it's much more powerful. So in this case, I have a list of substrate concentrations here ranging from 1 to 100 micromolar. And then I say, this is a for loop. So I start with the phrase, with the word for number in substrate concentrations, which is this list. we go through, we calculate the velocity, and then we print the velocity. So if I execute this cell, I get the velocities for all of the substrate concentrations that are listed here. And if I go in and I can change values and do things to that to see what's the impact going to be. So this is just some of the simple things we can do in that particular notebook. I wanted to move on now to another notebook, another lesson, which is about creating plots. So this is using data from our linear regression of protein assay. And in this particular one, the question here is, how can I plot and annotate my data in Jupyter notebooks? And so the objective here is to once again do linear regression, and then to create a plot of the data with the best fit line, and then to create a plot of the data that include confidence intervals. And one of the things you have to do in Jupyter notebooks is the notebooks come with a lot of tools built in, but if you want more tools, you have to, and more functions, you have to import libraries. So in this particular cell, I'm importing a library called OS, so that means this notebook will work whether you're in Mac or Windows or Linux. Pandas is a data management tool. So you import pandas, and then we're going to import stats from SciPy, which is a collection of scientific Python functions. And that stats function enables us to calculate the parameters for a linear regression. So if I execute this cell, then it goes through and it calculates actually the slope, intercept, R-squared value, p-value, and standard error for the data that I imported in this particular case. And I'm not going to take the time to show you how I brought those data in. And once we have this information, this is helpful, but we'd really like to have a plot of the data. And so what I'm going to do now is use a tool called, or a library called matplotlib to plot this data. And so here's a simple plot of the data with our absorbance on the y-axis and our protein concentration on the x-axis, but it's not annotated. We don't have the equation and we don't have a best fit line. So the next thing I'm going to do is I'm actually going to introduce a best fit line. And so now we have the best fit line based on the analysis that the Python code did. And you can, by visual inspection, it looks like a pretty good fit. And now I want to go through and annotate that plot a little bit. And so I can do that in this cell. And you can see that I have the x-axis with protein concentration in milligrams per mil, the y-axis in absorbance at 595 nanometers. And there's actually the best fit line. The equation for the line is shown here. And so that's really nice. And I was working with that and happy with that. But then I thought, I really want to show confidence intervals. And so I asked a colleague of mine, and what he suggested was, in fact, to use a new library that I wasn't familiar with from Python called Seaborn. So if I go in and I use the Seaborn library and I do the same kind of plot, I get a best fit linear regression, least squares fit here, but it also includes the confidence intervals with this particular type of plot. And so those are just some tools that are available for plotting the linear regression data. The final worksheet I want to show you, or notebook I want to show you, is nonlinear regression. So in this one, we're actually going to fit some Michaelis-Menten kinetic data to a curve. And in this particular case, we have to import a bunch of libraries again. In this particular case, we're going to import a library called, or a function called curve fit. And that function actually enables us to fit data nonlinear, or to fit data to any equation, any function we create. All right. And this is, I'm going to show you actually a little bit of markdown here. You can see in here we've got a couple pound signs here. That tells us that this is going to be a header. Then we've got simple text here. And then you look here and you'll notice I've got double dollar signs before and after this. This tells us that it's actually an equation using a markup language called LaTeX. And when we do that, we get this beautiful version of the Michaelis-Menten equation shown here. So what I'm going to do now is just do a quick data import. And so here's our data. The first column here, this is the substrate concentration, paranitrile phenyl phosphate. And the last column are the initial velocities that we calculated. All right. And so I just want to do a plot of these data. And when I do this, I can see that we have the kind of data you would expect for a Michaelis-Menten type enzyme. It's a rectangular hyperbola, nice, and we have the initial velocity on the y-axis and the substrate concentration on the x-axis. And so now what I want to do is actually want to fit a curve to this, do a nonlinear curve fit here. So what I'm going to do here is first create a function to calculate rates for Michaelis-Menten enzymes. And so MM is the name of that function. And then I'm going to use the curve fit function that's available from the SciPy library to actually determine the Km and Vmax. And so if I do this, if I use the curve fit function, you can see curve fit right here. I realize I'm running through this very quickly. In the workshop, we go through this one step at a time. Then I just fit the data to the curve, but I didn't plot it out. We need to extract the information from the optimized parameters. That's the p-op variable, and the p-cov variable contains some error data. And so I can actually extract the Km and Vmax from the curve fit. And so this is, once again, Km and Vmax determined from the data. So this is a nonlinear fit. And now I want to plot the data containing that, containing my actual data and then the fitted curve. And you can see that here. And note, if you notice the fitted curve, it fits nicely, but there's the only data points in the fitted curve are at the known substrate concentrations. I can actually use another program called linspace that's available. And if I do that, I can get this lovely fitted curve, very smooth curve that fits the data. So those are the Jupyter notebooks. And I'm going to stop sharing that and go back to sharing my PowerPoint. Maybe not quite yet. Just a second. Need to bring my PowerPoint back up. There. All right, so the future plans that I have are to offer live workshops in person or online, and to record the workshop I've developed on YouTube, and to develop more modules for biochemistry and molecular biology education. And if you're interested in these at all, you can contact me at this email address, paul.craig@rit.edu. And as I said, I'll make this available to the group. And then here's a list of a few resources that you can draw on. And I think I've gone over my time, so I'm not going to go through the resources other than to mention the Molecular Sciences Software Institute website here, which is a place that has lots of resources. And with that, I'll close my presentation. Thank you very much.
Thank you so much, Professor Craig. So
You're
once
welcome.
again, we've been exposed to new resources, and I bet those who already have some experience in coding should really take advantage of this programming. Thank you so much. And once again, on behalf of the organizing committee, we would like to present you with a certificate of appreciation for taking the time to come and present the example of using these Jupyter notebooks and educating the participants today. Thank you so much. Okay, so having heard the experiences and perspectives of online teaching from the Americas region, let's now look at examples from the Asian and Oceanian regions. So first, I would like to introduce our next speaker, Professor Elizabeth Johnson. Professor Johnson is the Deputy Vice Chancellor of Education at Deakin University in Australia. She's an Australian National Teaching Fellow with awards for learning and teaching from the Federal Government's Office of Learning and Teaching and from the Australian Society for Biochemistry and Molecular Biology. She is a Principal Fellow of Advanced HE. Professor Johnson has over 30 years of teaching in biochemistry and life sciences and has led national projects in university science education on scientific inquiry, quantitative skills, learning leadership, and work integrated learning. She established the Teaching and Learning Center of the Australia Council of Deans of Science. And at Deakin University, Professor Johnson's portfolio includes Deakin Learning Futures, which, among others, delivers Deakin's online learning environment and supports course development and building staff capability for learning and teaching. Professor Johnson will be talking about new techniques in biochemistry and molecular biology education in the new normal. I welcome you, Professor Elizabeth Johnson.
Thank you very much. It's a great pleasure to be with you here today. And of course, I will now share my screen so that we can work through the presentation together. And I hope that that's now clear. I am grateful to the organisers for inviting me, and I have very much enjoyed the presentations so far. The ingenuity and creativity of my colleagues is always impressive. I'm talking to you today from Melbourne, Australia, from the lands of the Wurundjeri people of the Kulin Nations, who are our local First Nations community. Although my background is in plant biochemistry and cell biology, I've been working at the centre of universities now for about 7 years. And so my remarks today grow from my experience across a wide range of disciplines. Of course, my heart remains with STEM, as you wouldn't be surprised to hear. Globally, universities have risen to the challenge of pivoting to online learning, as we've been hearing today. And this was desired to keep our students progressing through their study. And along the way, we've challenged our own assumptions about what makes excellent learning and teaching. And for today, I would like to get you to stand back from the detail with me and to think about digital learning for our future. And I hope to persuade you that digital education is not an add-on, it's the core to the way we work and live. So the first point to make is that our world is actually digital. As you've been hearing today, not only do we learn digitally, we work and study online. These are some headlines from around the world looking at what happened during the pandemic, and we moved our work online. We kept working, we just worked online. And that's not without controversy. As the last headline says, working from home is overrated. We've all had to juggle work and home requirements. We've met lots of other people's pets and families on the way through. And that's because the controversy is about adjusting our perceptions and our expectations to working online. But in the process, what we've done is become more digitally fluent. We've moved our own skill set towards a digital world. So I've brought together some ideas here about digital fluency. In the top left of the slide, you can see the triangle from the Digital Literacy Development Framework, old work now from 2010 from Beetham and Sharp, but it shows that shift in perception from knowing about things, access and awareness, to the point where they become normal tools for your everyday work, through building your skills to normal practice to your identity. You are a digital worker. On the right, you can see some comments there from the UK Digital Skills Task Force, which estimated that over 80% of future jobs would require digital skills, either as digital citizens who were using digital tech or digital workers who were evaluating or configuring digital tech. And we've just heard a beautiful example of that on talking about the Jupyter notebooks. The number of our graduates who will be able to get jobs with no digital skills required is very small indeed. So we need to build their digital skills, not just so they can learn in a disrupted environment, because that is their future. And while we think about that, we should be thinking about the digital skills which they will need for their future. And to do that, we need to understand where those skills sit. Machines, this work comes from a colleague, Margaret Bierman. Machines are better at following rules, at classifying and recognizing patterns, and they predict and inform on the basis of those patterns. Humans, on the other hand, we're the ones that set the rules, and we're much better at providing rationales, explaining patterns, speculating and interpreting. That is, we're better at investigating the why and machines are better at the what. Now, our exposure and use of digital learning was certainly accelerated by the pandemic. One small silver lining from this global pandemic is that it has done this acceleration, and in the process has forced us to accelerate our assumptions. And here's what happened. We moved from on-campus learning in specialist and generalist spaces through workplace learning over to sending all our students to work from home. And this change was emergency remote teaching, as described by Associate Professor Dries in his keynote this morning. And that shift meant that we were working at speed on an emergency response. So a little different to what we would do normally. Emergency remote teaching, as defined by Hodges in the reference that Associate Professor Dries gave you, is well-planned online learning experiences that are meaningfully different from courses online in response to a crisis or a disaster. And that, like all learning, effective online learning results from careful instructional design and planning, using a systematic model for design and development. That is, the things we're talking about now take time. So we shouldn't judge a premium online learning by emergency remote teaching. It's breaking down our assumptions and giving us a platform to grow from, but we have a lot to do to build out what it will look like for the future. And we can think about that in terms of how learning is designed. And I'm going to take you back to some work from a number of decades ago. This was work that came from Michael Moore, published in 1989, but it talks about learning as a set of interactions. So in the top left there, there's the interaction between learner and content. And a lot of the lovely things that we've seen today talk about biochemistry and molecular biology content. Learners can access them physically in a laboratory or I've shown a lovely library here. I do like libraries, but really most of our content is actually online. If you think about where biochemistry is published and shared, it's online. On the right, there's the interaction between learners. And again, a number of our speakers today talked about how important it was to create a learning community so learners could learn from each other. Now, we typically think of our learners as doing this on campus in small groups or in classrooms or in lecture theatres, but they can do it online, and they're often quite adept at it, more adept than you might think. Remember, many people interact online anyway. That's what social media is built on. And while I wouldn't want my learners to be doing their learning necessarily on social media platforms, that concept of online interaction is a familiar space. So the third interaction that Moore pointed to was the interaction between learners and instructors. And while we appreciate and enjoy a face-to-face interaction there, again, like the peer-to-peer learning, it can certainly happen online. Online looks and feels different, but it does have some big advantages, and here's three. It can operate asynchronously, that is across time zones, and allowing for other commitments that learners might have. If some of your learners are part-time and study and working at the same time, asynchronous interaction, for emails a great example, allows them to talk to colleagues at the time and place that suits them best. Which brings me to the next point, which is about geographic constraints. Online interaction simply removes them. And the third one, I'll just pop back to that last slide there, is that it stores records of interaction. So it allows learners to review back the interactions that they've had in the past. We can capture that as discussion noteboards or as chat. I want to reframe the idea of digital learning through two lenses for you. And the first one here is about accessibility. And we've talked a lot about this morning. And this really focuses on the replacement of on-campus activity. You can use it. We've talked a lot about online labs, but there's many other tools that you can use as well. And it offers some particular additional affordances relative to online activity. So on the left there's the idea of repetition to mastery. If you're providing online quizzes, the example I've got here is from the Khan Academy, your learners can revisit those quizzes, go over the material indefinitely for as long as suits them. The idea of online laboratories, and a number of beg your pardon, a number of speakers this morning have talked about the value of using simulations, not just as a replacement for laboratories, but complementary to them. So learners can develop their skills before they end up in the physical laboratory. And the artificial intelligence assisted learning allows us to guide students independently of an instructor. Remember, the AI is built by the instructor, so it's still informed by the instructor's knowledge of learning. These kinds of replacements for on-campus activity are complemented by the digital library and independent learning tools. But there's another aspect to digital learning that I really want to emphasize, and that's the idea that digital learning is authentic. It's about real world activity. It's not just a substitute, it's actually how we work. And if you think about that in terms of exploration, the first thing we reach for when we want to investigate something new is a search engine, whether it's an academic search engine, I picked Google Scholar here, but there's any number of them, or whether it's just a simple search engine where you're searching through what's actually happening in the real world. That is a real world activity. Analysis, as I'll reiterate later, for biochemistry and molecular biology is absolutely digital. There's no way we could do things like systems biology in any other way. And that is now increasingly true for visualization. I remember the first time I saw a 3D image of a molecule leap out of the screen at me. It absolutely changed my view of what molecular space looked like, and I'm sure it does for your learners as well. Communication is authentically online, as we are proving today. Flexible collaboration is authentically online, again, as we are proving today. And information management certainly is. So we can think about on-campus learning as a complement to online learning. So these two activities complement each other, and you can draw them together to make a really premium learning experience. Of course, there are things that we can do in a physical space that we can't do online. And the example there obviously is laboratories. Some laboratories are online, interestingly, and we'll come back to that a little bit later on. But for biochemistry and molecular biology, a lot of our work is actually done in a physical laboratory, and we want our students to experience that. That's also true of fieldwork and clinics, so specimen collection or field observation. And it's also true for performance. Although you and I are interacting online today, we would have a different feel if we were actually in the room together. And often people will say Zoom is good for transactional discussions, but it's not really good for the brainstorm or the critical discussion that you want to have with your students. So these are things we're now thinking about. What are the affordances of being online or being on campus? And I would point to the different types of skills that those activities produce. I'm going to run through some digital learning tools here, not in the specific. We've had some really wonderful examples of that in this morning, but to point out categories of tools for you. So you can then judge other tools for yourself and think about how they might come together in your learning design. And so we're going to whip through these 4 categories. First of all, a few comments about adaptive learning. So adaptive learning is an online pathway for learning, a guided pathway. So you might know examples such as Smart Sparrow is one that comes out of the Australia originally and now out of the US. What those programs do is they scaffold a learning pathway. The one I've given you here, the design I've given you here, talks about exposing the students to content in the first instance so they can become familiar with it, then spending some time on some mastery exercises, whether that's quizzes or whether it's writing exercises or other sorts of tasks, then scaffolding the student through, getting them to walk through to a test where they're testing their skills and knowledge. And at that point, there's an intelligent step that comes through. The outcome of the quiz then diverts learners either to new content, to revise practice, or to go back to revise concepts. Now, to set this kind of learning pathway up, you need to be an experienced learner in your discipline. You need to be able to understand where students will get challenged and where they'll get stuck so that you can design the learning pathway appropriately for them. So you need to be able to have at your fingertips the curriculum mapping and understanding of threshold concepts that will help you to design this well. These learning pathways are useful, but they do take time to set up. So my suggestion is that you focus them on those learning concepts where you want learners to develop their own picture of the learning and where they're likely to need reiteration and practice. The second group here is really about simulation. We've looked at some lovely ones this morning. So I've pulled out 3 categories here. On the left is interactive media. And the reason I put this up is because it's actually quite simple to set up. This particular example comes from my own university. It was used for a first-year biology class, and it's a simulation of a trip to a coastal ecosystem. What the teacher has done here is taken an image and embedded an interactive link into it. That's what the little plus signs are. And this is simply done through open sourceware. The one that we use is called H5P. You can download it fairly simply. And all it does is take the student then to a particular image taken from that point in the ecosystem, and they can do some analysis on that image. And that's a simple and accessible thing for staff to set up for themselves. The more sophisticated version of virtual labs, as has been pointed out this morning, there are open source versions of this. They can be very sophisticated, very complex, and very deep in their understanding of what students can do. Because they take time to set up, try to make them reusable. Try to be able to set up something which you can substitute new activities into, or you can use it in a number of different places. My own university does use virtual and augmented reality on the right there. And I've shown you 2 images. The top one is from our engineering virtual reality lab, which has sensors for both sight and touch, that is haptics. And so students can bring up an image of a molecule, for example, and walk around it and move it and use pressure to play with it. The one at the bottom, the augmented reality, is an image of a heart that was created so students could explore that in real time too. Both these activities require specialist equipment, so you do need to be able to invest in them. But they are using elements like gamification to engage students and to invite them into a new world. They help out with inaccessible and dangerous environments. And because the students can repeat them on their own, they improve practice to mastery. And we've heard about preparation. Analysis and visualization are not new to biochemistry and molecular biology. They're absolutely core to the way we research and teach our students. Most of our instruments now have digital outputs, and they send that digital output to analytic programs. So our students quickly learn that instead of using pens and paper, they're going to use digital tools for analysis. So I think incorporating this into our practice is absolutely a reflection of our daily research work. The other thing I want to point out is one other small silver lining from the pandemic, which is the progression to using digital visualizations in everyday language. This visualization comes from Worldometer, but similar visualizations have appeared on our newsreels for the last year. And I'm really hoping that that emphasis on data remains with us as a normal part of everyday life. We mustn't forget about the workplace tools which underpin all of this. We don't write anymore, we type. We don't draw anymore, we take pictures. We communicate by text, chat, blog, websites, team channels. Our workflow is managed digitally, either through workflow processes in places like SharePoint or project management or even intelligent automation. We use online to build large, diverse virtual communities, and online allows us to store and share information and manage security and privacy. These underpinning affordances, these underpinning tools should be what our digital learners are working with all the time, because they will be expected to be familiar with them when they move out into their future careers. There are two tools that I would like to bring to your attention, which are really peculiar to science. One of them is about remote experimentation. And the example I've given here is a particle collider, the Australian synchrotron, which is in Melbourne, my hometown, but also CERN, which is the Hadron Particle Collider in Geneva. Both of those experiments are virtual for the large number of researchers that work on them. The Australian synchrotron has beamlines that are available for remote learning. So physics students or structural biology students in Australian universities can connect to the Australian synchrotron and work there. I think the other exciting one is citizen science, and Foldit was mentioned in the plenary earlier today, a beautiful, beautiful option for students to go out and solve puzzles collectively. So all of that brings us to the idea of rethinking learning design and delivery. How do we bring tools together to get to the best possible outcome? And I want to start with the idea about what is good learning. And this is actually quite well understood now. Learning is most effective when it's active, when it's constructively aligned, and backwards design is an excellent example of that. Constructive alignment simply means that the objectives and the activities and the assessment reinforce each other, so they're visibly connected to students. Works best when it's engaging and relevant and when it's intentionally designed and evidence-based. This information is from the education literature. It's not controversial. It's the reality of how learners learn, and it's the reality of how adult learners learn. We've got to remember the why. What are we trying to achieve? Of course, we're trying to achieve the desired competencies and skills, but we want our learners to go beyond that, to be lifelong learners. So they need to develop independent evaluative judgment so they can judge the work of themselves and others, and their capacity for lifelong learning. Here, I've brought together a spectrum of online learning through to on-campus learning, because I want you to start thinking about how you put things together. And that spectrum of blended learning, which incorporates both on-campus and online, varies from very minimal support for face-to-face learning. Most universities in the world now use learning management systems or virtual learning environments to provide online sources for their learners through things like flipped classroom or mixtures of synchronous and asynchronous classes. An example of that is HighFlex, where the design is to have both online and on-campus options for learners at all times. Through online guided instruction, where the activity for the student is primarily online with the occasional input of the facilitators or teachers, right up to independent self-paced learning. So that spectrum offers a great deal of variety, which then can be tailored to your time, your students, your space, your objectives. And that's the important thing. All these tools are valid in their own right. You've got to find the ones that are right for your students. Which brings me to my final thesis, and that's that blended learning is the optimal result, and it's the final result for online learning. In my own university, 30% of our learners learn exclusively online. We are a 65,000 student university. We're big. That's about 12,000 online learners that are learning with us. And some of those learners are learning in science, not as much as in other disciplines, but certainly some. So it's not that online learning cannot stand alone. It's that you need to choose the learning activity that suits your intent best. Online is particularly good at repetition to mastery, at self-test, at confidence. It's particularly good at exploration, managing and sharing information and data collection. And it's good with structured thinking processes, whereas fieldwork, in-person, physical learning is better in different ways for experience, for observation, for data collection, for experiencing special environments, and for particular sorts of collaboration. So depending on the learning outcomes and the discipline and the authentic practice in the discipline, the end game is to choose a mix of these activities which suit your curriculum best. So I hope I have persuaded you that digital learning is real-world learning. Digital learning is digital work is authentic to work and life, and it will be increasingly so. Digital does increase access and participation, but that's not the only reason for using it. The reason that I keep in mind and I take to my staff all the time is it's because digital learning is the learning of the future, and that's what we need to focus our learners on. Digital learning comes in many formats and it keeps evolving over time, so it's important to interact as we're doing today with people who are bringing new opportunities forward, but they are based on the same good learning principles. So my advice is combine modes and tools to get the best outcomes that you can and personalize to your own environment. And I will stop there. Thank you for listening.
Thank you so much, Professor Johnson. So do you think this combination of face-to-face and online teaching would be a challenge for low and medium income countries?
Actually, it's more accessible than you might think. So the two big barriers to online learning are people's expectations. So interestingly, in my own university, that 30% of online learners were very comfortable during continuing study during the pandemic. They just continued with what they already had. But our on-campus students were initially very concerned, very anxious, and they lost confidence in learning. But that has been rebuilt over the last year. So managing expectations is a crucial part of it. The other challenge is around digital access and digital disadvantage. We do need students to be able to connect online, and Australia has great big areas of digital disadvantage because it's a big and rather empty continent. So that is an issue. But when you think about the degree of mobile coverage around the world for mobile phones, it's probably more to do with how we ask our students to use bandwidth rather than it being a real barrier. There are many, many tools which are easily accessible and simple for people to use, and connecting with communities like this will help people to see those tools and bring them in. My advice is don't commit to expensive licenses and big commercial production without understanding why you need it and why it's of value to you.
Right. Okay, thank you so much. And again, the organizers would like to show their appreciation for you by presenting to you this certificate. Thank you so much, Professor Johnson.
Thank you very much.
All right, so we come to the last speaker in session 1, and I'd like to introduce to you Professor Thilo Hagen. Thilo graduated with an MD from Humboldt University in 1996, and he subsequently completed various postdoctoral fellowships at Harvard in the US and at GlaxoSmithKline and University College London in the United Kingdom. He joined the Department of Biochemistry at the National University of Singapore in 2007, where he is currently an associate professor. His research focuses on energy metabolism and cell signaling. In his teaching, he is particularly interested in developing new ways to introduce problem-based and self-directed learning and in imparting transferable skills. Today, Thilo will expand on his experience in making online learning more fun and effective. Welcome, Thilo. Thilo, can you hear us?
Okay, give me one moment. I just had to change my laptop because for some reason I got disconnected from the Zoom, but I'm ready.
Sure, no problem.
Okay, 1 second. Okay, so I'm good to start.
Yes, yes,
Oh.
you can do the screen share now.
Okay. So about 1 year ago, I received the following email: teaching remotely, join Eric Mazur for an interactive webinar on remote teaching. Having heard a lot about Harvard physicist and pedagogist Eric Mazur, and facing the prospect of having to actually teach online in the coming semesters, I was really desperate to join this webinar, and luckily I managed to secure a slot in the last session here on Friday at 4 p.m. Boston time, which actually translated into Saturday morning 4 a.m. Singapore time. So I had to set my alarm very early on that day, but I can tell you all that attending this webinar was really worthwhile because it was an amazing experience. And before I tell you more about this webinar, let's first hear from the man himself, Eric Mazur, about what is peer instruction using learning catalytics and how it works in a live lecture setting. We have some error here. Okay. So I don't know. I have so many technical problems today. Not sure why, but let's start this again. Okay. Let's share the screen again. Okay. Right.
Professor, not able to see your video. We are able to see your board.
Okay. Wow. Let me just stop the sharing. Okay. So let's try this again. So share screen. Thank you. So how about now? It works?
Yes.
I'm not a big believer in high stakes examinations. You look at a picture of students sitting in an exam and you can just feel the anxiety level, the stress. Stress is not conducive to learning. Stress leads to cramming, to shortcuts, to trying to reach a good result with a minimum effort. That doesn't mean I eliminate assessment. There's lots of assessment. And one of those forms of assessment is the Readiness Assurance Activity, RAA.
So the RAAs are pretty much the equivalent of like a quiz or a test in this class. As with a lot of other parts of the class, there is an individual portion and there's a group portion. So it's a 2-hour assessment. So for the first hour, you're working individually on the 9 or 10 questions. For the second hour, they give you time to work with your group and re-answer the same questions, but as a team. It's the time for your teammates to share their knowledge with you if you're confused about a question, or it's the time for you to explain concepts to them.
When we launch into the team round, that's when the magic happens. The students see on their screen what each of their team members answered. And now they have to come up with a team answer to those same 10 questions. So I'll go, hey, I have 5 Newton here. Sarah, why do you have 7 Newton? And we might go to the board and one of us will go, oh, I forgot about that. And then once we at the table reach agreement, one of us enters that value into the system. You look at students in RA, they're smiling, they're learning. They're learning because they're having these aha moments during the team phase. they'll go, yes, we got it right, and high five. So you see a level of energy and activity that is just unimaginable for a standard assessment technique.
The emotions that a lot of students are feeling during the second half of the RAA are a lot more positive than I would usually associate with the test.
Sorry, can you please mute yourself? Sorry. Can we mute everybody, please? Sorry, can we mute everybody?
Yes, we are muted.
Okay, thank you. Sorry.
Associate with the test taking experience. You can hear students like getting questions right and like audibly celebrating, or sort of making these like frustrated sounds if they get it wrong. But because it's a group activity and you have the support of your team, it's much less nerve-wracking and it's a lot more fun.
Yeah, so for me, there are really three main advantages of team-based learning. Firstly, learning through teaching others usually has much higher retention rates than most other forms of learning. And I know this from my own experience. If I really want to know about a new topic, the best way to do this is by actually teaching this topic to students. Secondly, as teachers, we are often far above the clouds. What I often experience is that students can often explain things to each other much better than we can. And that's partly because they share a common background, a common foundation, but also because students usually have just managed to understand a concept, and therefore they can actually explain to the other students, to their peers, how they managed to understand it. Whereas we as lecturers usually don't remember how we initially comprehended a concept. And lastly, thirdly, team-based learning really makes online teaching much more engaging. And in fact, what I have found is that through using team-based learning, online teaching in large classes can actually be more engaging than live class teaching. Which brings me back to the webinar that I attended last year, in which we attendees and lecturers were actually the students. We were asked to answer a set of questions individually, and we were then put into breakout rooms where we discussed these questions again and tried to come up with a joint answer. And what happened in these breakout rooms was really quite amazing. I was together with 3 other lecturers from different parts of the world, and being able to see what everybody had responded in the individual round, we actually started discussing immediately. And when we then came up with a joint answer and submitted it, and it turned out to be right, we really felt immediately connected. So it was really an amazing experience, and I wanted to carry this into my upcoming Year 2 cell biology module. And after implementing this over the past semester, I have realized that there are 2 important aspects that are required to make team-based learning and peer instruction successful. The first is the type of questions that we ask. And the second important aspect is the use of a suitable platform. And those are actually the 2 things that I want to talk to you about today. And we'll start with the questions. So of course, we all know that if we ask students knowledge or fact-based questions, they will usually try to bypass their brain and use a very well-known search engine to come up with the answer, and the learning outcomes are very limited. And the students, in fact, also don't like this approach. And I had to experience this firsthand when I started teaching some 10 years ago and got feedback that usually looked something like this, right? In this case, out of 156 students, 81 students felt that I was delivering too much content, and 34 students complained that there was too much memorization in my module. And I must say, at this point in my teaching career, I felt quite demoralized and depressed, because there I was delivering or giving what was probably pretty poorly delivered lectures with content that the students could have easily found, in textbooks or online. And I must say, I really questioned my existence and justification, as a lecturer. So it felt really uncomfortable. And I was thinking to myself, what can I offer the students that I am good at and that the students cannot readily find in online sources? And what I eventually decided I would do is to actually teach the students how to interpret cell biology research data, because that was something I was good at. And it was also something that requires a lot of practice and that students therefore cannot really find online. And as I thought about this more and started implementing this in my teaching, I realized that this approach actually has a lot of advantages. Firstly, being able to interpret data and ultimately understand research papers is really a skill that every science and life science students should have. Secondly, when using research data, it is actually really easy to come up with problem-based questions, which normally it's rather difficult if we teach content and facts. In order to make time to actually discuss research data in my lectures, I actually had to cut a lot of content. So for instance, instead of teaching maybe 5 to 10 signaling pathways, I would only discuss 1 or 2 signaling pathways. But I would discuss them in very minute detail, including discussing the methodologies that students need to know in order to interrogate these signaling pathways. And as a result, students actually have the tools now to study and learn about other topics by themselves. Yeah, so in a way, we have enabled the students to learn independently. And so instead of spoon feeding them with information that they forget most likely, we have given them the tools to actually even access primary research papers to get the information that they need to know. And finally, by using research data in my year 2 module, I also make it clear to the students what is the process to which researchers arrive at scientific conclusions, and show them the notion that many scientific problems remain unresolved and that we actually need them to help to solve these problems in the future. So we all know, of course, that in order to have a successful course, we need to make sure that our teaching is well aligned with our assessment. And in order to create this assessment, what I have done for many years now is to base my midterm test and my final exam on a research paper that is related to the content of my module. For instance, in the past semester, for the midterm test, I used a research paper on apoptosis. For the final exam, I used a research paper on the cell cycle. And I give these papers to the students well in advance. And then during the test or exams, I will ask the students questions that are related to the figures of the research paper. Now, you might ask, what kind of questions, right, do I actually ask the students with regards to research data or figures of a research paper? So I want to give you an example, right? So this is, for instance, the figure 1 of the midterm test apoptosis paper that I used in the past semester. And I would use 3 types of questions. Firstly, I might ask the students to interpret the findings. Secondly, I might ask the students to predict the results that they would get if, instead of using a DNA fragmentation assay, in this case, they would actually use a Western blot. Yeah, so I would in fact ask them to draw out the expected results, right? So basically place a band of the Western blot in the provided Western blotting panel. So you can see already, right, in order to answer this question, right, the students need to understand the method, they need to understand the results, and they have to apply this knowledge to actually predict the results. And the third type of question that I would ask is, I would change some of the conditions in the experiment and ask the students to predict what the result would be. For instance, I would say, what would happen if we knock out a certain protein that is involved in this experiment, or if we add a certain inhibitor, and then ask them to predict this result. So there, and over the years, I mean, I realized that there are a lot of advantages of having this kind of final exam. Firstly, it is really easy to come up with new questions every semester because I'm using a different research paper. Secondly, when using research data and research papers, I can actually come up with real problem-based questions that are multiple choice or multiple answer questions. That is something that is usually quite difficult to achieve when you teach like knowledge of facts, right? So that's another advantage. And thirdly, you know, for these year 2 students, they really feel a sense of achievement at the end of the semester when they are able to read and understand a scientific research paper. And they are likely much less hesitant to, in the future, actually access primary research literature by themselves. As you have probably realized now, in order to make this approach work, it is necessary that the students understand the research methodologies that are generally used in cell biology and that are specifically used in these research papers. And to address this, I have a set of videos that I let the students watch outside the lectures, which describe the critical methods. And then we use the lecture times to actually apply these methods to specific research problems. Okay. So now I would like to move on to the platform that I used and that I mentioned is really important when trying to implement team-based learning. So I used Learning Catalytics, which was originally developed by Eric Mazur and colleagues at Harvard. And this platform, which is a web-based platform, is ideally suited, you know, for peer instruction because it was designed for it, and it allows you to deliver question multiple times. And I made full use of it in my course over the past semester by literally using it in every class that I taught. And in this course, there were 100 students, which I divided into permanent teams of 4 students per team. And then I had 2 types of activities. In my Thursday online lecture slots, I used a format which is called instructor-led synchronous question format, where students respond individually to questions as they are delivered one at a time. This format is not so different from clicker or poll everywhere question that many of you are familiar with, except that I usually delivered the question twice, right? Once where students answered the question individually, then I would send them to the breakout rooms where they then discuss and then re-answer the same question. The second format that I use on Mondays is the team-based assessment. And this is the format that you have seen in the video at the beginning. Here, the students respond individually to a set of questions again, and then gather in their groups and respond as a team to the same question where they try to come up with a joint answer. So the way I implemented this was I would come up for each lecture with a set of 8 to 10 questions, which the students then are given 25 to 30 minutes to first answer individually during the lecture. And I will send them then into the breakout rooms where they rediscuss these questions and come up with a joint answer. And as you can see, right, I awarded either participation marks for these assessments, or I actually graded these assessments in some of the lectures. And I really like this type of assessment because there was a lot less stress involved, and the students really learned during the lecture, because I mean during the assessment, because immediate feedback was provided. We are actually going to have a demo later on during the large class parallel session on the team-based assessment. And therefore, I want to now quickly show you how I actually implemented the research data-based individual question format in my Thursday lecture. So we're going to watch a short clip now that illustrates this. And now we want to confirm that MAO is indeed in the outer mitochondrial membrane and only in the mitochondrial membrane. So what we do now is actually we subfractionate the mitochondria into the four compartments that we have just mentioned, right? And then we load protein samples of all four compartments on our Western blot and blot for MAO using a MAO antibody. We also use VDAC, which is actually a marker for the outer mitochondrial membrane. Based on the Western blot results shown, our question is now, does MAO likely only localize to the outer membrane or likely to both the outer and the inner membrane, right? And so this is our next question, which you can try to answer. So somehow I lost the presentation here again. Am I still actually audible, or? And now we want to confirm that MAO is, sorry.
Yes, yes.
I'm still audible.
Yes,
okay,
yes,
so I see. Okay, that's great. Okay, right. So for some reason, the video, and now we want to confirm that MAO, okay. Anyway, so as you have seen, I explained the question, then stated the question. And now what I would actually do is actually open the question in Learning Catalytics. What you couldn't see actually for some reason that I actually used a different laptop to open the question, which is easier, but you can also do it from the same laptop from which you're giving the presentation. And then I give the students some time to answer this question individually. And during this time, I actually can see on my screen, right, how many students have responded. And based on this, I can decide how much more time I want to give the student. And I can also see how many of the students got the answer right? And based on this, I can decide whether I want to include a team round in this question, which I wanted to do in this point, in this case. So let's have a look now. So 30 more seconds. Okay, 5 more seconds. Let's have a look at the results. It's not so clear this time. Let me show this first. So yeah, so here I'm actually showing the results after the individual round without revealing the correct answer, because I want to include a team round. We are going to actually go into our team round for this, so you can discuss it again together. Please choose the breakout room that corresponds to your team number. Then we have about 4 minutes. Yeah, okay, and then I will send more seconds. Okay, then I will send the students to the breakout rooms. We deliver the question, and then the students will answer the question again. And as you can see, after the second team round, there is really a marked improvement in the answer, right? So now most of the students actually got it right. And this was, of course, not an isolated case, but it was quite a common phenomenon. And that was obviously really exciting for me to see. Now, let me share some of the feedback that I collected at the end of the semester. I have had individual research database questions for a long time, so I was mostly interested in finding out how useful the students found the team discussion component. And as you can see here, the majority of the students found it really very useful. And so that was good. But one potential problem with team-based learning is that within a team, there may be one student who is very dominant and does all the explaining, and who may provide all the, you make all the decisions in the team, and then there may be one or more students who are very passive and don't really contribute to the discussion. And therefore, I was interested also in why the students found the team component helpful. Was it because the explanations by my fellow students helped me to understand better, or because explaining things to my fellow students helped me to understand better? And very reassuringly, 71% of the students felt it was because of both of those, which suggested that most of the students really actively participated by both explaining and getting things explained. Now, I want to come back now to the team-based assessment, where the learning catalytics is really, really useful because it has a number of features which really make this work really well. And the first of these features is actually that the students in the team round, in the breakout room, can see the answers of their team members, so from the individual round. And this is really important because it allows students, when they put into the breakout rooms, to start discussing immediately, rather than having to go around and ask everyone for their opinion. Also, some students may not want to actually voice their opinion. They may want to wait for others, to first, say what they think, and then they just agree with it. By showing all the responses of the team members, we can ensure that the opinions, right, of all the students contribute to the discussion. And again, when I asked the students, how important was the feature to be able to see the individual answers of the other team members on your screen, almost everybody thought that this was a very important feature. Another very important feature of learning catalytics is that there is immediate feedback. Yeah, so the way this would work normally in the team stage, the students will come up with a joint answer, submit this, and then if the question answer is correct, the system will tell them so. If the answer is wrong, they will get the following response. Sorry, your response to question 1 was not correct. You can attempt this question 2 more times. And then once they have used up all their attempts, then they will get the following message. Sorry, your response to question 1 was not correct, and you have attempted this question the maximum number of 3 times, which is 3 in this case. And then the correct answer is shown along with the explanation. So as you can see, there really is, in every case, immediate feedback. Of course, the students can also go back later in the semester and review their inputs as well as the explanations. But the point is that the system provides multiple feedback and the ability for students to answer multiple times. And again, when asked, all the students felt that the ability to answer or attempt a question multiple times is really important. And this brings me to the end. And at the end, I would like to ask you whether I have convinced you really that using research data-based questions and peer instruction is really a useful way to make online lectures more engaging. For me, myself, I can say that it definitely was. It was more engaging than any other live module that I have given in the past. And it has not only resulted in my best feedback scores so far, but it has also really made teaching a lot more fun to me. And I suspect it has also made learning more fun for the students. And with that, I would like to end. If you have any questions, please feel free to email me at this email address, bchdh@nus.edu/sg. And with that, I hand it back to the session chair, Sheila. Thank you.
Thank you so much, Thilo. That was very interesting. And I'm sure your last question, you might actually find it being a discussion point during the breakout sessions, the parallel sessions later. Okay, so again, on behalf of the organizing committee, thank you very much for taking time out to come and talk to us. And for that, we would like to present you with this certificate of appreciation. Thank you, Tilo.
Thank you.
So I think you will all agree with me that this has been a very informative session, and we thank all the speakers for sharing their expertise and also for sharing their resources to the participants. Thank you so much. And now I hand back to Ashwini.
Right.